Yes, thank you, Anna. Welcome also from my side to this GHGA lecture today. So we have Peter Krawitz with us today. I will give you a bit of his background. Peter started physics and medicine in Munich. And he also, during this time, he stayed at the Institute for Systems Biology in Seattle and did some research on Boolean networks in the framework of his diploma thesis in physics. He obtained a medical doctoral degree from the University of Munich. And after that, he went to Berlin and worked as a clinician scientist in medical genetics at the Charité and as a postdoctoral research fellow in the group of Stefan Mundlos and Peter Robinson. In 2016, he became a group leader of clinical bioinformatics at the Charité, the Institute for Medical and Human Genetics. But soon later, he moved to Bonn in 2017, where he is, until today, the head of the Institute for Genomic Statistics and Bioinformatics. Even way before ChatGPT and the current hype about artificial intelligence started, Peter has been interested in how artificial intelligence can be used to support the diagnosis of rare diseases. And as a result of his research in this field, he developed several tools for this purpose. And one of these tools he will present to us in particular today. This is GestaltMatcher and its data portal. Welcome, Peter. I'm happy that we have you today, and I pass on to you and look forward to your talk.
Yeah, thanks a lot for inviting me to speak in that round. I know only some of you personally, so I hope I can get you interested in our work. And I will start sharing my screen. So I'd like to comply with the housekeeping rules that you just announced. So I understand that questions will be asked in the end. And therefore, I will rather try to keep it shorter so that we have more time for discussions in the end. And then maybe I will search for appropriate slides to visualize my answers. Anyhow, so since some of you are not familiar with the algorithmic ideas of GestaltMatcher, I will spend some time on explaining those ideas. And then later on, I will motivate why we need it in clinical genomics and what we need from GHGA. Okay, so some of you are familiar with Face2Gene. That's an app that many clinicians now use in their work, but also pediatricians, particularly for them, it's handy because they are good at recognizing whether a kid is looking funny. I mean, that's even a term used sometimes in clinical notes, but they are not very good in coming up with differential diagnosis. So this is a tool that can support pediatricians or clinicians with differential diagnostics of dysmorphic kids. And for the interpretation, you need to have a deeper understanding of the technology. And I will explain in the next few slides what similarity scores are and how to use them. In the end, we do pairwise comparisons of portrait images, or basically medical imaging data from any kind of domain. We could also do it with X-rays from hands or fundoscopies. But in the end, it's always a pairwise comparison of these data sets where the image belongs to one case. So we have a matrix of all the individuals that we compare. And if we think about a single case, we are interested in the similarities to all other cases that we might have already solved. Then we are talking about one vector. Or if we basically want to do cluster analysis, we are talking about the matrix, right? And there are use cases for both. Okay, and in the end, I can use these values from the confusion matrix or distance matrix also to compute distributions. So let's visualize that in 2D. So every individual is now a dot in that, on that screen. And I can measure basically the distance in between. And if I plot the distance just on the x-axis, this will result in distributions, right? And perhaps the distribution looks bimodal. Then it's more likely that this is an overlay of two different distributions, right? And what could that be? That could be same syndrome, different syndrome. So that's the hypothesis, that whenever there's not just a smooth normal distribution, bell-like shape, that this is an overlap of different distributions. And now let's talk about the interpretation of the scale. So we are measuring similarity. We can in the end discuss whether similarity of healthy individuals is the same as similarity of affected individuals. But in the end, they are very related. So monogenic twins or self-self comparisons will be very close to the origin. And siblings that share a high degree of ethnic similarity, because they are from the same parents, are also closer to the origin than individuals from very different ethnicities. And then somewhere in between, we will have comparisons between individuals with the same disorder and comparisons between individuals with different disorders. And same disorder is closer to the origin than comparisons between different disorders. So keep that in mind. The lower the Gestalt score, that's how I will refer to it now, the more likely the individuals share the same genetic cause. Okay, and this is how it actually looks like, right? So you get the idea on the left-hand side, and on the right-hand side, you see the distributions for a same syndrome comparison and different syndrome comparisons. And the first thing that you acknowledge is that on the right-hand side, it really looks smooth, right? But this is also what we should expect. Zentraler Grenzwertsatz, large limit theorem, and basically everything will look like a normal distribution. And we also have lots of such comparisons. So in the database, we have roughly 10,000. So we are talking about 10 times 2 comparisons here. And for the intra-syndrome comparisons, it looks a little bit more bumpy. But if you think about it a little bit longer, this is also what you would expect, because there we have different degrees of distinctiveness, right? So we do comparisons from Kabuki with Kabuki, Cornelia de Lange. These will be very much on the left-hand side. And syndromes that are less characteristic in the phenotype, like Turner maybe, they will be more to that, closer to that threshold. Okay, and that is basically the question of lumping and splitting. I will come back to that in a little while. So lumping is, do the phenotypes look similar enough to merge them in a phenotypic series? Here, an example of two individuals with Marbury syndrome that are caused by genes mutations in different genes, but in the same pathway, all participating in synthesizing the genes, the enzymes of the GPI anchor deficiency. And here, on the other hand, I just took an example of individuals that share mutations in the same gene, but it's a pleiotropic phenotype. More about that later. So here we would split. So what's the working hypothesis? So you all know the saying from Bauhaus, form follows function. What we are trying to do is the reverse. We are trying to delineate dysfunction from dysmorphism. So it's not la pour la. We don't want to do the lumping and splitting just out of fun. If there is evidence, if we think that there is evidence for splitting, that means they don't share the same cause for their disease. It's a different molecular mechanism that's causing the phenotype. Okay, and is that approach justified? Is there hope that we can delineate such information from image information? And here I would like you to think a little bit about the complexity or the information that you can encode in different domains. So you all know how large the genome is, and you have a rough idea about the numbers of combinations that can cause monogenic phenotypes. But now do the same about images. And just as a much simpler image, I printed a QR code here. So if you Google about QR codes, you will also find the question, will we run out of QR codes, right? But if you do the math, you see that is not going to happen. There are so many combinations for QR codes that it's more than atoms in the universe. And in a portrait photo, the state space is even larger. So sure, a face needs to adhere to certain constraints. You need a nose, you need eyes, you need a mouth. So we can't get rid of that. But still, there's so much room to encode information that in principle, it should be possible to deduce dysfunction from dysmorphism. So how do we do it? The main challenge is basically to get rid of all the noise or the information that's not clinically relevant. And how can we do that? So basically, that it's possible has been shown by all the papers published in that field. Now let's quickly have a look at the recent advances. So I'd like to talk a little bit about when it started. Basically in 2015, that was the first time a deep convolutional neural network was used for dysmorphic work. And then later on, there were advances on the architecture which further improved the performance. And that's basically work from PhDs in my lab and other labs that I'm presenting today. And let's quickly go through the app. It lists likely diagnosis, and it does a good job when it's a relatively common rare disorder, but it didn't work so far for the ultra rare ones. So I just take as an example here one case where there were just 2 solved individuals in ClinVar, right? This is how the plot is organized. So I ordered the disorders by the number of submissions in ClinVar. And if I want to match this individual with a case in the database, I can use only a network that wasn't trained on this disorder. So on the right-hand side, you see the disorders we used here for training. So this is all parameters that were already trained on normal faces for face recognition or intraperson verification. Then I added a last layer for the fine-tuning on rare disorders. And now I work with the penultimate layer, the feature layer, as a coordinate in my clinical face phenotype space. And this is where we started, right? Now we have the distances again of the portraits in this high dimensional space. And if they are close, they are likely to share the same cause. Okay, what influences the performance? It's the network architecture. And if you can update the network architecture, this might also result in a improvement of performance. And Alex had a very smart idea. He basically acknowledged that if you train if you have enough cases to train a model for a specific disorder, that's what you should do. But if you don't have enough, then you do the classical GestaltMatcher approach and do the matching in the clinical face phenotype space. And this is called an ensemble approach. He basically combines the best of both approaches. And just look what that does to performance. So here in the 22 paper, we still worked with the very old architecture. I will show that again on the timeline a little bit later. And frequent is basically we have more than 10 individuals to train the model for a specific disorder, and rare basically means we have less than 10. And this performance, so the readout is always how often do you see the correct diagnosis in on the top position, or is the correct diagnosis among the top 10 differential diagnoses. This is the accuracy readout, and it improved from 16% to 53. Or from 20 to 36. So this is an incredible improvement in performance based on the same training data, just by improving the architecture, right? So, and this is the improvement from one year to the other, basically. Okay, let's talk a little bit about the similarity that I now mentioned so often. If I ask in that audience, do you think the kid of my, so my brother has twins, dizygotic twins, and if I would have a poll now, does Victor look more like the dad or the mom? There is no clear vote in the audience. And this, there might be two different interpretations. Either the audience is very incompetent, or it's a stupid question. So I tend to think now that it's a stupid question. And this is also what most clinicians now do. They don't just say, this looks like Kabuki syndrome. They try to be more specific about the features, right? So they try to name the abnormal findings. It's highly arched eyebrows that are laterally rarefied. So this is a very good description of a peculiar finding in that syndrome. And then you maybe could also answer that question of my brother's kids. Maybe the eyebrows look more like those from the mother or from the father. Okay, and I mean, in a clinical context, we also use that for naming disorders sometimes. Okay, I will skip that a little bit because you can do that at home afterwards. So is the AI in the end more objective than clinicians or humans? I would argue it's not, because the AI also depends on the training data and the algorithms, the architectures of the network. This will always result in slightly different answers, but I also don't think that it's a bad thing, because maybe those questions that are difficult to answer always go along with some subjectivity in the reasoning. Here it's cooler in a live audience. I'd like to know your opinion, what you're looking at. I give you a shortcut. It's basically dysmorphologists looking at dysmorphic faces, right? We track their eyes. And you can see that they all look at the eyes and the nose intensively, but still there's a lot of variation. And even if the measurement is varying, then probably also the assessment is. Okay, and just a quick comment again on these timelines. So here on the lower part, I try to visualize what we use from consumer electronics. I mean, these are hundreds of great computer scientists working in computer vision, and we benefit from it, right? So this DCNN, deep convolutional neural network from 2015, basically resulted 2 years later in the first Face ID from the iPhone. And 2 more years later, it was the first algorithm used in face-to-gene. Again, this technology improved a lot after 4 years. A new architecture was published, ArcFace, and this is also the architecture that Alex used recently in the ensemble approach, right? So this also explains improvements in the performance because an entire industry is working on it. Okay, and if you want to know what the faux pas from Google was, it was in the beginning object labeling where the engineers didn't take care of diverse data sets. That's why an African guy was labeled as a gorilla. But nowadays, this can still happen because it's very hard to do a good diverse training set, right? It's more important to know why this happens than that it happens. And I think this is also what we have to be aware of when using AI now, they often just try to circumnavigate this problem, right? I mean, sure, it's a problem, but it's more important to understand why that can occur. The challenge of learning on very limited data will go along with compromises on this political correctness. That's basically what I want to say. And we simply have to acknowledge that we also have to learn on very limited data. And sometimes it works very well. So for example, here, maybe you have never seen these two species, but I just show you two examples, and all of you will correctly answer that on the right-hand side, it's a pangolin. For other phenotypes, it might be more difficult. So this is a pig, this is a sheep, this is a cat, this is a lion, this is also a lion, this is a shark, this is a dolphin, this is a bird, and this is a turtle. Incorrect, but not really wrong, right? So it looks a little bit like a turtle. And here is another interesting aspect. We are currently working on a collection of syndromes that a medical illustrator or caricaturist is generating. And if we can recognize a person by a very compressed data set, basically, I can store the caricature with less data than the original image, right? And we can still recognize it, then this is compression or improvement of signal to noise ratio, which we can also use again. And the AI, by the way, will also still recognize it. So a second component on how to deal with the rare data problem. Okay, and one last comment on the subjectivity, objectivity. So we acknowledge that humans are subjective in their assessment. AI is subjective in their assessment too. And a way to visualize that is also by using explainable AI approaches. And there is not one solution. On the left-hand side, I list a few methods of generating attribution nets, and the results look different. But it's very hard to tell what's preferable. And maybe, again, it will also depend on the clinician, what he or she prefers. Okay. Yeah, this is, I skipped that. Okay, now let's talk again how we do clustering in the similarity matrices. We basically compute phylogenetic or the similarity here. And I already showed you these two earlier. Basically, they are similar to a prokaryoid phenotype, but they still form a distinct cluster. And in the end, they also have another pathomechanism. And that is what we now can contribute computationally to that task force. And here I give you maybe 1 minute so that you can try to do the same. It's a pleiotropic phenotype. All are caused by mutations in SMARCA2. But in one disorder, the eyes look different than in the other. And GestaltMatcher perfectly separates that. So this is an example where it works tremendously well. So the phenotype of the same disorder is very much on the left-hand side from this lumping and splitting threshold. And if I would compare these two groups, we would have a mean score very much to the right-hand side. Okay, very much. And we can do that for everything that the task force worked on, and it works in a very high percentage. And just keep in mind, not all of these phenotypes look different, right? So a pleiotropic gene can also be a non-facial phenotype. Okay, and we can also do that now on new genes, right? I mean, the proof of concept is on everything that has been published, but here it also worked very well for a new disease gene, and it's very suggestive visually because it also splits in exon 1 and 2. It's only a gene with 2 exons, and mutations in exon 1 nonsense mediated, go to nonsense mediated decay. So it's a loss of function phenotype. And in exon 2, they escape NMD and it's a gain of function. Okay. This can also be used as a, sorry. 1, 2. We also used that in the Translate-NAMSE study, which was a project preparing also genome medicine. So the future data we want on GHGA. And in Translate-NAMSE, we had a very representative set of cases. This is basically an overlay with all the OMIM disorders. So most of them had a neurodevelopmental phenotype, but that is the same in OMIM. And we could solve a comparable number to UK studies. And again, we can speed up the analysis with using the Gestalt scores, but there's more use of the Gestalt scores than just improving the efficiency. It's also a data quality control. And for that, I have a good example. Here we have a girl that was presented several times. And the subjective diagnosis was colon de Vries very much from the beginning. But single gene sequencing didn't result in a diagnosis. Exome sequencing didn't result in a diagnosis. And the Gestalt scores were so indicative that we did a genome and looked in that region. And by that means we could find and solve the cause. It was a small deletion that just involved a few base pairs of the exon. That's why we didn't detect it with exome sequencing. And it escaped MLPA. And in a genome, it was simple, right? But you had to get to that level where you really had good arguments to go for a genome and to look into that region. Okay, and I just show that because I'm annoyed that this paper gets rejected for so many times. I think the data is more convincing than what DDD published 100 times repeatedly. We had more interest, we had an enrichment of ultra rare disorders, which was really clear also by the more recent availability of these diagnoses. I mean, what do you publish as a disease-causing gene in 2020? Probably it's an ultra-rare disorder, right? Our friends in England wouldn't have had to sequence thousands of genomes. They could have done that with panels too.
Peter?
Yeah.
Sorry, I think we have to come to an end to give the audience also the opportunity to ask questions.
Ah, okay, sorry. Then I basically stop here and take all your questions about the FAIR data now.